All right, it is my pleasure today to introduce Dr. Greg Carr. He is an assistant professor at the Lieber Institute for Brain Development and also at the Department of Pharmacology and Molecular Sciences at Johns Hopkins University. Dr. Carr is a little different than some of the other interviews we've had before. He's a person that's a little bit out there in the trenches trying to discover new drugs for people to actually help them, not just understand the basic brain circuits and all this kind of ivory tower stuff. He's actually trying to help people with new compounds, which we desperately need for a bunch of disorders. And so he's particularly, he's kind of a rising leader in the field in pharmacology, drug discovery, especially with regard to schizophrenia and other disorders. And he's especially interested in cognitive processes and how they relate to these disorders. And he'll be talking today, at least sort of revolving around his paper published in Psychopharmacology in 2020, novel non-nitrocatechol methyltransferase, or COMT, inhibitors modulate dopamine neurotransmission in the frontal cortex and improve cognitive flexibility. Welcome, Dr. Carr.
Steve, thank you for the introduction, and thank you for this opportunity to talk with you about this paper we have. It's a paper that we're really proud of in my group, and I was really excited that it was in psychopharmacology because psychopharmacology has a special place in my heart. It was the journal that published sort of my first primary research paper. I was second author on a paper back in 2008, and we've been fortunate enough to publish a few other times in psychopharmacology. And so really excited that this paper made it in, and really excited to talk to you about it today.
Fantastic. Could you tell us a little bit about why you're studying COMT and how it relates to schizophrenia? What's the role of, what is dopamine doing in schizophrenia? Whatever it is you think would be most interesting as far as background for understanding your paper.
Yeah. So I'll start. I'll try and touch on each of those points, and I'll start with the fact that in the paper we're describing these novel COMT inhibitors. But the enzyme was discovered way back in the late 50s by Julius Axelrod at NIMH, and so it's been studied for a long period of time. And there have been inhibitors in the literature for many years. And in fact, there are 3 of them that are FDA approved for clinical use. So they're used as adjunctive therapies for the motor symptoms associated with Parkinson's. And so the enzyme COMT is one of the enzymes that metabolizes L-DOPA in the periphery. And so you can co-administer a COMT inhibitor with L-DOPA. It increases the amount of L-DOPA that makes it into the brain, where it can then be converted to dopamine and improve those motor symptoms. And so one of the things that got us interested in it from a psychiatric perspective and from a cognitive impairment perspective is that COMT also metabolizes dopamine in the regions of the brain, like the cortex, where there's very little of the dopamine transporter. And so because there's very little dopamine transporter, COMT is essentially the major regulator of dopaminergic tone in these regions. And so this mechanism, COMT inhibition, had been proposed as a mechanism for treating cognitive impairment, or for treating symptoms where there may be sort of a hypodopaminergic tone. And so this idea had some sort of clinical proof of concept from the Parkinson's literature, actually, where tolcapone, which is a COMT inhibitor that's brain penetrant, it's able to improve both the motor and cognitive symptoms associated with Parkinson's disease, whereas peripherally restricted COMT inhibitors only improve the motor symptoms, essentially through their interaction with L-DOPA. And so there was a lot of excitement about this, but unfortunately there were some severe safety concerns with tolcapone, particularly around long-term use. Essentially, it can produce fatal liver toxicity. So it's really not a viable strategy to use tolcapone for sort of these long-term chronic indications like cognitive impairment associated with schizophrenia. And so what we thought was, okay, there's a need for a safe brain penetrant COMT inhibitor that'll allow us to test the hypothesis that COMT inhibition is a viable strategy to combat cognitive impairment. And so, in this paper, really what we're doing is we're describing for the first time sort of the in vivo effects of these novel COMT inhibitors that we developed here at the Lieber Institute. And the key pieces for us with these compounds were they're structurally distinct from the inhibitors that are currently on the market. And they're also selective inhibitors of the membrane-bound form of the enzyme. And so we and other research groups have shown that the membrane-bound form is the one that's responsible for metabolizing dopamine at physiological concentrations. And so the really nice thing is here we can selectively target dopamine metabolism with having little effect on the soluble form, which is really important for sort of the detoxification functions of COMT in the periphery. And sort of the major findings in this paper, where we show that our lead COMT inhibitor produces similar effects in the brain with respect to dopamine metabolism and behavior, as you see with tolcapone. Specifically, we show the accumulation of the dopamine metabolite DOPAC and depletion of HVA in the brain and CSF. And this is important because COMT converts DOPAC into HVA. So for us, this was evidence that we're inhibiting the enzyme directly in the brain, where what we think would be the site of action for cognitive impairment. And we also show improvement in cognitive function using a set shifting assay that had been previously shown to be sensitive to COMT inhibition. So all in all, we thought this is a really good starting point for a drug development project. And we're hoping to see this advance into clinical trials here in the near future.
Oh, very cool. It's exciting to be so close to drugs that are actually going to be going into people that they might actually improve their quality of life.
Yeah, definitely.
And I'm wondering about with regard to the downsides of the prior generation of COMT inhibitor drugs. You said that there was some significant toxicity with repeated use. Are you looking to see whether this drug has anything like that as well?
Yeah, that's sort of ongoing work at this point. So normally what we'll do is we'll try to nail down the efficacy, so to determine, we're getting the desired effect. And then from there, we kind of work through sort of the undesirable effects. And there's a pretty sort of, really well laid out plan that the FDA requires for you to show with your compound that you're not having, either effects on cardiac function, liver function, genetic, what we call genotox, so genetic toxicity, mutagenesis, those kinds of effects. So there's a sort of standardized assays that these compounds will go through. And we're hopeful that we're able to have a nice sort of clean safety profile alongside a significant efficacy profile.
Fantastic. And do I understand you correctly that you're proposing that this drug could be useful for either Parkinson's or potentially cognitive effects in schizophrenia? Is that right? Cognitive deficits, I guess.
Yeah. So with this COMT mechanism, we really think this is sort of diagnosis therapeutic, right? So we're really thinking of it as indicate what we call clinical indications. So diseases that are characterized by difficulty with executive function, working memory, attention. Those types of symptoms cross a number of disorders, including schizophrenia, Parkinson's disease, ADHD, even potentially sort of disorders characterized by impulse control, substance use disorders as well. So we really think, this is kind of a symptom-based approach where it may have efficacy across a number of disorders characterized by, sort of these deficiency in frontal mechanisms. And so that's kind of what we're going to be looking to test. If we do have a compound that makes it into the clinic, is, what's the best clinical indication, or is it something that's useful for many?
Interesting. So it's it sounds like you're kind of taking the NIMH recommended RDox kind of strategy here, where you have like this transdiagnostic symptoms essentially that may sort of be present in a bunch of different disorders as defined by DSM. And so, I mean, I think that's a really interesting approach. I'm really glad to hear that in the drug discovery field that you guys are kind of thinking a lot about this as well, because I can say in the basic neuroscience field, this is definitely something that's been on my mind a lot. So could you say anything about that approach? I mean, compared to prior approaches, for example, do you have any sense?
Yeah. So I can talk a bit about how this fits into the drug discovery work that we're doing. So we really think about two different distinct strategies for drug discovery. One is, you try to figure out what is disrupted in your indication or in your disease, and then you directly try to either reverse it or prevent it from happening, right? So you're going directly to the cause of the disorder and trying to remedy that. And so that's what a lot of sort of the work around the genetics and the environmental risk factors associated with, in our case, schizophrenia, but for your disorder of interest, a lot of that work is designed to help you either intervene before a patient has reached sort of pathological levels of the disorder, or to reverse sort of those pathological changes. Another strategy is to really take the system as it is once a diagnosis has been made and identify where you have sort of deficiencies or issues with functioning that people would like to improve. And there it can be agnostic to how the system got there, right? So you don't necessarily need to directly address the underlying problem. You can come at it from orthogonal directions, angles, and say, okay, we know this particular mechanism improves this particular function. So let's see if we can augment that and get a therapeutic benefit. It may not address the disease itself, but you can get some therapeutic benefit for patients, improve their quality of life. And so really, that's kind of, we take the sort of two-pronged strategy there. And the second strategy, where we're looking at sort of symptoms or trends across diagnoses, looking at symptoms and affecting systems that way, it's really helpful when you don't know the exact cause of the disorder. And so in the case of schizophrenia, we're still learning about how a person develops the disease and how they get to a point where they're diagnosed. And so until we have complete information there, we think it's also valuable for us to use this symptom-based approach.
Absolutely, we got to help people as soon as we can, even if it doesn't necessarily cure them, and at least if it makes them feel better in any way. That's important work as well. So I love this two-pronged idea. That seems to me like a very promising way to go forward.
Yeah, exactly.
Great. And so can you speak a little bit to how you got interested in this type of topic? Like, what is the nature of your interest in COMT and in these sorts of disorders and drug discovery?
Yeah. So I think going back to graduate school for me at Penn, where I worked with Dr. Irwin Luckey, kind of working on novel mechanisms for potential antidepressant therapies. I've always been interested in sort of this translational space, using preclinical models to help identify potential therapeutics that may have benefit in the clinic. As a postdoc, I transitioned over to schizophrenia-focused research and really kind of honed in my work on the cognitive impairment associated with the disorder. And the reason for that was, it was a major unmet need. Sort of the therapeutics didn't do a good job of addressing those symptoms. And we know from the clinical literature that quality of life and functional outcome for patients is directly correlated to the severity of their cognitive symptoms. And so I thought, if we're going to have, the biggest impact we can have is trying to figure out how to improve these domains for patients. So working as a postdoc, still sort of behavioral pharmacology, but now focusing on cognitive impairment. And when I started my lab here at the Lieber Institute, both the CEO of the Institute, Danny Weinberger, and the head of drug discovery, Jim Barrow, had extensive experience working with COMT and sort of its mechanisms. Danny, from sort of a clinical perspective, and Jim, from a medicinal chemistry perspective, actually trying to design inhibitors. And so I think it was a natural starting point for the Institute in terms of the first drug discovery project that we really dug into. And so given what we thought would be possible if we had a good drug that could target this mechanism, plus the expertise that the Institute had coming into it, it seemed like it was a really good place for us to start.
Yeah, makes a lot of sense. What would you say is the biggest outstanding question or sort of next big step that we're going to make or need to make in this kind of line of research?
I think, the main, I think, outstanding question, and we touched on it a little bit earlier, where, you were asking, so, what disorder do you think this is going to work with? Is it going to be sort of a cross diagnosis? I think the next big question for us, and this is something that we're working with, with a partner now at this point. So we've partnered with Boehringer Ingelheim on this project for the next stages. And one of the big questions for us is, which clinical indications, which disorders should we go after first? Which one do we think it's most likely to benefit? And I think there's a case to be made for a number of disorders, from schizophrenia to Parkinson's, to really address the cognitive symptoms of Parkinson's, to potentially substance use disorders. We think there's an element of improving cognitive control with this mechanism that would be possible. And so I think the biggest outstanding question is, which indication is the best one to, or I should say, which one do we think would have the largest effect in for patients? And that's still an open question. We're doing some experiments to try and help us come to a decision there, but it's definitely still open at this point.
Great. Well, it's exciting work, and I really look forward to seeing how these drugs pan out in the clinical trials that you say are going to be starting. Thank you so much, Dr. Carr, for your time. This has been a great conversation. I really love the perspective, a little different than mine, working at the sort of edge of industry and the edge of translation for sure, but in animal models. And thank you also for your contributions to psychopharmacology.
No, thank you for the opportunity to talk about the paper and this project. Something we're really excited about. And I always love the opportunity to interact with Psychopharm. As I said, it's been a scientific home for me and my work for a long period of time. So I really enjoyed the opportunity.
Outstanding. Thank you so much, Dr. Carr.
Thank you.